So the sparrow, they are primary consumer and secondary consumer. So when it eats the seeds, fruits and peas, and then the sparrows are primary consumers, and also they can act like the secondary consumers, that is the third tropic level when it eats the insects and worms. And in the diagram, like where the producers are the first tropical level plants, that is phytoplankton, grass and trees, they can produce the biomass through the photosynthesis. And it depends on the and then there is primary consumer second tropic level, that is herbivores, that eats the grasses, the zooplankton, that animals that eat, mostly eats the phytoplankton, then grasshopper that eats the grass, and then the cow eats the grass. And then secondary consumers, that third tropic level, that carnivores that eats the animals like the birds, in which the bird species and wolf. And the tertiary consumers, that includes the woman and lions, that fourth tropic level, that is top carnivore. So, in the functional unit of the ecosystem, it has the decomposition, in which the first step is the that is the fragmentation, in which the detritus like the earthworm break down the detritus into smaller particles. This is fragmentation. And bacterial and fungal enzymes degrade detritus into the simpler inorganic substance. This is catabolism. Detritus is a dead remains of the leaves, bark, flowers, and dead remains of animals, and including umbilical matter. That is the detritus, which is the raw material for the decompositions. And during mineralization, that is the humus is further degraded by some microbes, and release of inorganic nutrients occur by the process known as the mineralization. So this is it. So derivatives into simple. What is the bioprocess engineering? So it is the maintenance of the sterile, that is microbial contamination free. In the chemical engineering, and so that only desirable microbes eukaryotic cell can be grown in the large quantities by using the bioreactors. And in genetically modified organisms, there are three basic steps: that is identification of DNA with desirable genes, introduction of the identified DNA into the host, and maintenance of introduced DNA in the host, and transfer of the DNA to its progeny. So this is in the GM of organisms. Plasmids used as a vector. So the techniques of the genetic engineering, it includes the creation of the recombinant DNA, the use of the gene cloning, and the gene transfer. So that you can transfer the desirable genes, unlikely in traditional hybridization where the undesirable genes also get involved. Asexual reproduction, it preserves the genetic information. Sexual reproduction brings the variation. And there are origin of replication, they start the replication of DNA. And there are the clonings to form the identical copies of any template DNA. The first recombinant DNA was made by the Herbert Cohen in 1972. The plasmid taken from the